Although asthma affects bronchioles, most of its root causes are more inflammatory and thus involve the immune system. And they involve so many diverse aspects of the immune system, including aspects of innate immunity, such as the production and overactivity of eosinophils, and the inflammatory responses mediated by mast cells and basophils, and adaptive immunity, such as the production of Th2 T lymphocytes and the class switching of B lymphocytes to produce IgE antibodies. Upon exposure to pollutants, allergens, and other noxious stimuli, the epithelia, the smooth muscle cells, and the fibroblasts can release inflammatory signals, which then cause increased migration of various white blood cells from the blood vessel. They also then can help naive T helper cells mature into the Th2 lymphocytes, which promote inflammation. If epithelial cells are damaged, it can interrupt the barrier between the deeper bronchial tissue and the irritants and allergens in the airway, thus increasing access of these noxious stimuli to nerve cells, to white blood cells, and potentially increasing inflammatory responses. Dendritic cells can interact with the bronchial epithelial cells, both receiving signals from them and giving signals to them. Some can migrate to lymph nodes where they can help the maturation of naive T helper cells into the Th2 lymphocytes, which promote the inflammatory response. The Th2 T lymphocytes, they secrete a host of local hormones which have diverse inflammatory effects by promoting the maturation of eosinophils from the bone marrow, their migration to bronchioles, and increasing their activity, by promoting the inflammatory responses of mast cells and basophils, by causing the hypertrophy of smooth muscle, and by causing B lymphocytes to class switch and produce IgE antibodies. Eosinophil granule release worsens inflammation and can cause the hypertrophy and hyperplasia of smooth muscle, increases the extracellular matrix proteins, such as a collagen network being laid down, and alters the epithelium. B lymphocytes can undergo class switching, in which they no longer produce the antibodies IgM or IgG, but instead produce IgE. IgE binds to cells which have the IgE receptor, such as basophils, mast cells, and to a lesser extent, dendritic cells and eosinophils. This receptor allows them to interact with the allergen and then to undergo inflammation as a result of the presence of the allergen. The release of basophil granules can cause the inflammation, which results in remodeling the bronchiole. Also, it increases the migration of white blood cells to the bronchiole and may help immature T helper cells mature to Th2 cells. Basophils may even present antigens to T lymphocytes. When mast cells release their granules, perhaps after IgE has bound an allergen, this will cause additional inflammation, which can result in the remodeling of the smooth muscle of the bronchiole wall and can increase the migration of diverse leukocytes to the bronchiole. Given this complexity, it is clear that not only is asthma multifactorial, caused by multiple genes and environmental factors, it might also be asked, is asthma simply one disorder? In some patients, the eosinophils are primarily involved, which involves perhaps one set of treatments. In others, it is the IgE responds to an allergen, which is more involved. Some individuals have genetic predispositions, and there are a host of different environmental factors which can cause asthma to initiate or to worsen.